Hello everyone. Today, in the subject of industrial biotechnology, we are going to learn about disposal of laboratory waste. So, safe disposal of laboratory waste is very much important and essentially required in every laboratory setting. So, what will be the outcome of this lecture? What you will learn out of it? Particularly, this topic, you will be able to learn about how to dispose of the toxic waste. So, firstly, human activities produce a biological waste in the form of human excreta or other discarded materials of which may contain infectious microorganisms. So most of the time the excreta that is produced by humans or other biological waste and discarded materials, they what they basically contain. Basically, they contain organic material, and most of the time, these microbes are trying to break down these organic materials, right? That is why they produce a lot of odor and bad smell. Such waste, if untreated, has varying degrees of potential to cause diseases. So it is very much understood that such kind of waste, if untreated and left in the open, they could readily cause a lot of infection and diseases. Right. So it is the main concern of different organizations these days that you must dispose of the waste properly and in a very proper manner according to the protocol that have been designed by those particular countries. So, and it is a potential cause of death, disease and death also in lot of countries because most of the time this garbage is left unattended in the open and human activities all around it, and this generally causes what? It generally causes the disease to take a form of epidemic and pandemic and spread widely all around in that scenario. Existing methods of sanitation have served effectively to protect the public's health from any disease associated with biological waste. So, the as the conditions have been studied by the scientists and doctors and all experts all around the world. So, what they have concluded, they all have concluded the same um, points. And what the main point was, the most effective way to control such diseases or such communicable outbreaks of diseases is that to have a proper sanitation. Because 80% cases of these diseases will be reduced just to zero if the individuals are having proper sanitation facilities. So, if the sanitation facilities are effective and readily available, uh, they do generally protect the public's health from any disease that is associated with the biological waste, or most of the time, if the host is carrying such disease and his excreta is being straightly subjected to sanitation sewage, then no spread of disease will occur. Okay, and the life cycle of that very particular disease will be broken down immediately. So, our main purpose should also focus on breaking down the life cycle of that very particular disease. So, every time we should focus on these things, right? So, our understanding of the conditions required to prevent the transmission of disease has allowed the development of simple yet highly effective management techniques for handling biologically contaminated waste. So, most of the time, when the waste is biologically contaminated, when the waste is having all these contaminated things that are contagious in nature, so what are the preventive measures that should be taken by any individual? Most importantly, you must learn that if you are disposing of waste and excreta and all, contagious contaminants according to the protocol, then no possible danger is present there. Okay, so if you are disposing the things of in a very controlled manner according to the protocol, and you are treating the waste according to the protocol and sorting it off, then the waste is moving ahead and being disposed of properly in different facilities, then there is no danger of such pandemic or epidemic outbreak or any of the communicable diseases that can spread among the human population. Right? So it is extremely important for you to learn that every time these diseases they need to be uh, very much properly handled, and if handled carefully, then there is no danger of such kind of diseases. A brief review of some of the conventional measures used to protect the public's health are as follows. So, looking into this matter, the government has designed several guidelines and several protocols that should be followed in order to protect the individual from such diseases and such outbreaks. So, historically, human excreta have been linked uh, to outbreaks and diseases such as dysentery, poliomyelitis, typhoid, and cholera. So, when all such diseases were studied carefully by the experts and the scientists, what they all had concluded, they all had the very same uh, effect and very same result, right? They all concluded simultaneously that all these diseases are a result of uh, infections and not with disposing of the waste properly, leaving the waste unattended in the open, and defecating outside in open. Right. So because of defecation in open and a lot of things like that, what does it do? These things they readily transmit the pathogens to other humans, and because of that, the condition gets worse, and they left their feces unattended in the open. And from there, these pathogens they travel all around, and they can readily infect any human that is passing around it and that is present near to it. Such waste may contain high concentrations of pathogens that can contaminate food and water supplies also. Right? And because of these things, uh, it is very important so that you always develop a good sanitation facility. So, if no sanitation facility is available, and it is readily accepted worldwide that uh, high concentration of pathogens are already present in the waste, and they can readily contaminate the food and water supplies, because most of the time people they defecate near to the water body, and if rain occurs or anything like that happens, uh, the water that is contaminated with the feces will enter into the water supply. So this will readily complete the life cycle of that very particular pathogen, and it will infect everyone who is using that water for drinking purposes. Okay, to minimize the opportunity for such cross contamination, several fundamental principles of sanitation are applied. So these all things are under the category of cross contamination, like how you have to deal with these things, and how you should follow the principles so that this process of cross contaminations it does not occur and readily infect the healthy population. The first principle consists of providing a physical barrier. Okay, the very first demand is to provide a very basic and simple physical barrier and sanitary sewer system. Okay, the settlements they should have active sewage system, which consists of pipes and pumps to convey pathogen-laden sewage to a treatment plant. So it is not only important for having a sewage system or proper sanitation facility, but it is also important to have a sewage plant where they can turn this contagious sewage into a non-harmful form. So sanitary sewer system, which consists of pipes and pumps to convey pathogen-laden sewage to a treatment plant, effectively provides such a barrier. So what they are trying to do is to keep away this contagious species and contagious sewage away from the active human population. So proper facilities are required to be built, and proper functionalities are important so that people can understand the importance and learn the very basic hygienic principles. Once the waste reaches the sewage treatment plant or sewage treatment facility, other mechanisms help to reduce the disease cause, causing potential of the material. So different methods are applied for the sewage treatment facilities, which uh, what they generally do, they generally reduce the waste and all the material that is essentially important. And this type of material is subjected to a lot of treatment with alum and all other uh, procedures where the contagious nature of this material is reduced with the help of those treatments. The treatment of sewage usually consists of biological degradation of the organic material. See, all the sewage is having organic material, and that is why all these microbes are after this, after the organic components of the sewage. So they are what they are trying to do. They are trying to decompose the sewage material, and in the main process of decomposing, all the gases and bad odors are produced. The physical and biochemical conditions that are optimal for the degradation are often hostile, right? So the conditions are that are biochemical and physical. They are also very hostile to the survival of many human pathogens. So such conditions are made and such conditions are produced so that the conditions, the living of these dangerous host pathogens are very much difficult. The end result is significant reduction in the members of viable pathogenic microorganisms remaining in the treated waste. So the end result is significant reduction in the members of viable pathogenic microorganisms remaining in the treated waste. So what the experts and scientists are trying to do, they are trying to formulate the techniques and procedures which can readily reduce the effect of such things. Okay, the effect of the pathogens that are found in sewage system and all those systems. Right, because as a result of that, these things they are confined and they are killed and they are neutralized, and the disease-causing capacity of these contagious sewage is reduced to zero. And most of the time, it is very important and it is always discussed that humans they do have this significant facility available. Laboratory waste disposal summary. So make sure the materials placed in the municipal waste are suitable for the type of disposal. Okay, most of the time you should always classify the type of waste that you are dealing with. Okay, and municipal waste are suitable for the type of disposal. So you should always classify uh, the waste that should be uh, disposed of in what color of container. Do not place any liquids in the municipal waste. Okay, if you are going to place the liquid waste in municipal waste, it will be a problem. Do not dispose the chemical waste, including stock containers, uh, with unused products in the municipal waste. So, chemical waste should not be disposed in municipal waste, including stock containers and unused products in the municipal waste. Empty or rinse the containers must be free of any hazardous residue and be marked empty. So every time when the waste is being collected from the containers, they should always be free of everything, or there should not be any residue present in it. So and they should always be marked empty after the disposal and cleaning. All shafts must be in an appropriate puncture resistance container to prevent injury. So these sharp objects they should not be placed in these bags rather than in hard plastic containers. If a material can be mistaken as hazardous, radioactive, or biological waste, but it is not, it must be identified as non-hazardous. So, proper classification and sorting methods should be followed from the very early beginning. So, infectious potential of laboratory waste. So, what is the infectious potential of laboratory waste? To what extent the laboratory waste can prove itself to be contagious in nature? For laboratory waste to cause infection to anyone, six essential factors must be present. And what are those six essential factors? These factors are as follows. First, the presence of an infectious agent that is capable of invading and multiplying within a human host. So, the presence of an active agent, okay, agent is very active and capable of invading and multiplying within a human host. So, that very particular agent is fully capable of invading inside the human body, evading the immune system, and multiplying itself. An environment for the infectious agent that functions as a reservoir, allowing to survive and perhaps multiply. So, where these pathogens are generally present, they are present in a location of infectious agent where they all are living, and that very particular place is known as a reservoir. The reservoir is allowing it to survive and multiply without any uh, hesitation or without any danger. Third, a mechanism for the agent to escape from the reservoir. So, the mechanism through which this disease causing agent is trying to escape out of the reservoir. So, mechanism should be present. Otherwise, all the passages are multiplying in the very same place and they are not at all posing any danger to any situation. A mean a means for agent to invade and penetrate or enter the human host. So, when the agent reaches towards the human host, then it should have this speciality, then it should have this importance of invading inside the human body, penetrating it, and entering a human host. Six, a human host that is susceptible to infection by the agent. So, human host is also susceptible for the infection that is being caused by the agent. Risk to the general public's health. So, one must understand It is not only important so that we should learn the risk for the individual, but also for it is important and dangerous for the health of general public. Risk to the general public's health can be associated only with indirect modes of transmission because the public is not directly exposed to the institutional reservoir or the infectious waste generated by them. So, general public does not produce these type of infectious or contagious waste. They just produce kitchen waste. But the institutions that are running health facilities and industry, they use such contagious and harmful chemicals, and they also treat several other types of diseases. So they do have institutional reservoirs and infectious waste generated by them, and it should be dealt according to the protocols because general public has nothing to do with that. And if this type of waste is left untreated in the open, it does create a lot of issues. It does create a lot of problems, and it also lead towards the exposure of general public that has nothing to do with such type of waste. For indirect transmission to occur, the infectious agent must be capable of survival outside of the reservoir for an extended period of time. So, for indirect transmission to occur, we need an infectious agent or vector that is capable, infectious agent that is capable of survival even outside the reservoir for a very extended period of time. Okay, so the pathogenic agent is able to survive outside the reservoir also for a very long duration of time. There also must be an opportunity for the susceptible host to be exposed to the agent. So now the next step is that the susceptible host should also be exposed to the pathogenic agent. If the host is not going to be exposed to the agent, then it will not cause any sort of disease in humans. Right? Modern sanitation practice, as discussed in the introduction, minimizes the occurrence of such agents. Okay. So there also must be opportunities for susceptible hosts to be exposed to the agent. And modern sanitation practice, as discussed in the introduction, minimizes the occurrence of such agents. So, in all the cases, what you have to do, you have to avoid the general public for in order they get exposed to the contaminated and dangerous waste. So, our full-fledged responsibility is to uh, keep the general public away from such hazardous waste or such dangerous and contagious waste. A properly functioning community sanitary landfill, solid waste incinerator, or municipal sewage treatment facility provide adequate containment and treatment for infectious waste, even when the waste is introduced without prior treatment. So, proper waste disposal facilities should be present there, and they are always able to dispose the things off according to the protocols, and all the methodologies should be followed properly, and it should not pose any sort of danger to any human population or any sort of exposure to the general public present at that very particular location. Occupational risks. Occupational risk. Unlike the general public workers who generate, handle, and process infectious waste, have the potential for direct exposure to the infectious agent. So, biological waste haulers are the people who generally uh, are present on the front line. So, these workers they have the responsibility of handling and processing the infectious and contagious waste. And because of that, they are on the front, and they also have the dangers of exposure of infectious agents. Exposures can occur through direct inoculation, right? Uh, you sometimes you can get inoculated directly, uh, and how? Such as when a worker is accidentally cut by a piece of contaminated glass, because you don't know that what type of waste you are disposing of, and has it been autoclaved prior to being transported. And if accidentally a piece of contaminated glass cuts you, or through inhalation when handling process generates aerosols. So the workers who are working in the disposal of such type of waste, they should wear full PPE kits and carry full protective equipment against the exposures. Occupationally acquired illness associated with the handling and of infectious waste has been reported. So, occupationally acquired illness associated with the handling of infectious waste has been reported. So, it has been reported that individuals who do such kind of job, they sometimes they do get uh, uh, very degree of exposure, and to some extent they do, although they are adopting all the safety measures, they do get some sort of diseases, and these diseases are known as occupational hazards, or the diseases that they get on their occupation or because of their occupation. Protection against occupationally acquired illness is achieved through appropriate waste handling and treatment methods, which either contain the waste or uh, inactive the infectious agent. So it is very important to offer the protection to those individuals, because whenever they are working, they are exposing themselves on the forefront. So it should be our duty to minimize the potential risk for those individuals. Okay, so whenever you are disposing of the waste from your laboratory or from your facility, we should always autoclave it so that the individuals who are disposing of, they do not get exposed to such kind of waste. So, in this lecture, you would be able to understand and assist institution and generators in establishing prudent practices for the management of the waste, and especially management of the infectious waste. So, characteristics of infectious laboratory waste. What are the main characteristics, and how these things are different from others? Like, what are the things that make such kind of waste infectious in nature? Infectious laboratory waste is characterized principally as waste that contains microorganisms capable of causing infection in a healthy susceptible host. So these microorganisms, they are fully capable of causing infection inside a healthy and fit host. Once that very particular host has been exposed to such things, hospitals, healthcare facilities, medical research institutions, and industrial facilities can generate infectious laboratory waste. So, this thing I was telling you in the early beginning of this lecture also. See, the hospitals and healthcare facilities, including the medical research institutions and industrial facilities, also on the other hand, they do generate infectious laboratory waste. That is very dangerous in nature. Why? Because these type of waste, they generally have the infectious diseases treatment dressing, and syringes and the waste from the healthcare facilities, but also they contain some cytostatic and cytotoxic chemicals uh, from the industrial facilities, and these chemicals are very harmful in nature. So these type of waste, they not only have contagious agents, but also harmful chemicals with them. So, categories of operations that produce infectious waste include the following things. And what are those things? Operations that involve the processing and analysis of specimens for diagnosis, separation or purification of cells or substances from human blood. Okay, so they involve processing and analysis of specimen for the purpose of diagnosis, and separation and purification of cells or substances from human blood and body fluids, and in vitro and in vivo methods for propagation of pathogenic microorganisms. So everything should be followed up according to the protocols. Medical operations in which invasive procedures are likely to result in waste contaminated with blood and body fluids from an individual harbor an infectious agent. So you don't know anything about medical operations and other invasive procedures. Sometimes the individual is having disease, and sometimes he or she is not having any sort of disease. But you cannot predict it, and most probably, and most importantly, most of the time they are carrying contaminated blood and body fluids, and they do harbor a lot of infectious agents with them. So it is your utmost important thing that you are always careful about dealing with such things. Veterinary operations involving the study of zoonotic diseases in which infected animals, animal carcasses and tissues, contaminated fomites such as disposable instruments and supplies, and contaminated bedding materials are produced. So, not only human clinical uh, facilities and industries they do generate such kind of waste, but also when it comes to the dealing of veterinary operations and veterinary clinic, they do have dangers of uh, communicating zoonotic diseases that are transmitted from animals to humans. So every time these things are present, and they always do pose a lot of danger. Anatomical pathological services, where workers process specimens from individuals either known to harbor or who are at an increased risk of harboring an infectious agent. So, in pathological laboratories, what happens? They are most of the time collecting samples from the potentially infected individuals. Sometimes they might be having disease, sometimes they might not be having disease. So every time they have to treat every individual as if he or she is harboring that disease. So the blood samples and body fluids that are recovered or into the laboratory waste are also uh, at a very high risk. Diagnostic research and industrial operations that involve the collection and processing of bulk quantities of human blood, blood derivatives, body fluids, and the production of biological products in which pathogenic microorganisms are used, such as vaccine. So every time when you are coming in contact with blood derivatives, body fluids, or bulk quantity of tissues, and all these clinical waste, every time you should be extremely cautious. Okay, and you have to use precautionary measures for your own safety and safety for others. Here in this image, it is shown that how these zoonotic diseases are transmitted. Some are, especially these diseases are the diseases that are transmitted from animals to humans, right? They can be vector-borne, they can be airborne, proximity or coming into direct contact to animals like cats and dogs, food-borne that are transmitted from chicken and all those things, and their main aim is to infect the humans. So such diseases are known as zoonotic diseases. Biological waste is objectionable and putrescent. characteristics containing viable microorganisms that are either not known to be hazardous to humans or are minimally potentially hazardous is not considered infectious laboratory waste. So biological waste is with objectionable and putrescent characteristics. If biological waste is putrescent in nature, it is smelling bad because of the presence of all these organic materials that are in present in it. Okay, so because of these things, you cannot label that this particular waste is hazardous in nature. Humans or are minimally potential hazardous is not considered to be infectious laboratory waste. So examples include tissues or medical waste generated from the care of individuals who have not contracted an infectious disease. Solid waste including such items as soiled diapers, animal bedding material as per litter, animal carcasses, and garbage from the food processing plants and uh, and eating establishments. So all those things they are essentially dangerous in nature and they should be treated in accordance with the protocol. Objectionable non-hazardous medical waste is typically generated in extended care facilities and healthcare services. Adherence to good personal hygiene is proven. Uh, Food and sanitation practices affords adequate protection to individuals involved in the handling and disposal of this type of waste, particularly. So every individual must practice good level of personal hygiene and sanitation facilities should be made available to them and using and washing hands soap with soap frequently should be practiced. Handling and disposal of this type of waste is also essential. So, what are the responsibility for the safe handling of and disposal of infectious waste? The primary responsibility for the safe handling and disposal of infectious waste resides with the generator of waste. This responsibility extends uh, to the ultimate point of disposal, even when there are other parties involved in the handling of the waste. So every time you should make sure the waste is being disposed of, and if you are taking services of some kind of company or some kind of organization in the handling of the waste, they should do their job honestly and in a very responsive manner. The generator should contact inspections uh, to take other measures to ensure that waste is being handled and disposed of properly. Even though management of infectious waste is also the concern of waste haulers and treatment facility operators. So, generators of the waste should have this sorting facility where they can easily educate people to put what kind of waste in what type of dustbin. Because in a long term, it does generate a lot of headache for the waste disposal companies and haulers, uh, those who are working in the treatment facilities, because mixed waste is the most problematic thing to dispose of. In addition, there may be federal, state, or local regulations controlling medical waste disposal and record keeping must be observed. Every time proper auditing and record keeping should be practiced. Okay, and it should not be like that you are not keeping any record and handling things like that. So it is not important for your own safety but also for the safety of others. And according to the federal and state laws, uh, proper record keeping should be uh, done and it must be observed at all costs. It is very important when you are disposing and handling infectious waste. The major problem associated with infectious waste is the potential or for the occupational exposure. Okay, if the health individuals who are working 24/7 among the diseased individuals, then there is a lot of problems, and they do have some uh, occupational hazards. Okay, that is linked with the occupational exposures. The disposal of infectious waste should therefore be performed in an effective manner that minimizes the potential for exposure uh, of those by virtue of their employment must handle the material. So, if they are the waste holders or the medical practitioners, nurses, they are all coming into contact of these diseased individuals, and they are discarding of this such kind of waste. And they should always be attentive while handling these things. Right? It is incumbent upon the scientific community to educate the general public about effectiveness of current sanitation practices in protecting the public's health and to direct legislators' attention to the problems of occupational hazards that are associated with the handling of waste. So, scientific community should run the programs that are extremely important in educating the individuals and professionals how to handle such kind of waste and what are the precautionary measures that they should be taking and how to act in accordance with the emergency cases. Everything should be known crystal clear to the individuals and to the professionals who are working in such scenarios and such facilities, and they should know what are the occupational hazards and how they should handle such infectious and contagious waste according to the protocols that should be employed, that not only focus on the protection of individuals but also for the protection of waste disposals. Those are the individuals who are always exposed on the forefront of disposing the waste. Generators of infectious waste. The initial recognition of a potential hazard should set in motion uh, all of the mechanisms from social and final disposal, from the sources of final disposal. So, from the very early beginning only, the sorting out of different kind of waste should be practiced by every institution, so that the final disposal mechanism of infectious waste is much more easier and it is much more effective. Recognition is made most effectively by the generator of waste. Okay, the responsibility for proper handling, treatment, and disposal of infectious waste material resides with all those who knowingly come in contact with these materials. So, one must recognize that who is the highest generator of such kind of waste, and are the responsibility that they should follow while handling such kind of waste, and treatment and disposal of infectious material resides with all those who knowingly come in contact with these materials. So not only those individuals who are producing it and sorting it off, but also for the individuals who are treating it in the treatment facility and disposing them off should also remain safe from the potential hazard. However, the major responsibility for proper handling belongs to the initial generator of potentially hazardous material, because such material should identify and segregated according to its degree of potential hazard. Once the material has been identified as infectious waste, proper packaging and containment should be ensured until the decontamination or inactivation can be accomplished. No waste management program is functional unless all appropriate personnel are cogi- uh, cognizant of the aims uh, of the program and trained in the procedures for handling the waste. So every time institute should train their individual or the service people so that how they should handle the waste and how to use the basic protocols. This will handle a lot of difficult situation very easily and on a very grassroots level it will sort out the problem for the waste holders. The training program are developed and implemented. Training should be a continuous process. So every time the institution should run the training facilities for the servicemen. Haulers and waste treatment facilities. Infectious waste is often decontaminated at the generating facilities prior to its transfer to the disposal site. This is the part of main protocol. Whenever you are dealing with infectious waste, and it is the role of waste generator to sort do the sorting of the waste and after that to also uh, to do autoclaving before discarding the waste in the bags. Decontamination protects the waste haulers from the risk of infection. Okay, because when you are following the decontamination procedures, uh, these things they generally what they do they generally protect the howlers who are handling the waste. In those instances where the waste hauling company transports untreated infectious waste to a treatment and disposal site, adequate physical containment measures should be provided to minimize occupational exposures. The processing of infectious waste by treatment facility requires the same stringent attention to detail that is required of the generator treating its own waste. The treatment company has the responsibility for ensuring that all procedures are adequate and that all systems are functioning correctly. So both hauling and treatment companies should ensure that all personnel are made aware of the potential hazard of exposure. Personnel should be properly trained in all of the pertained aspects of the containment and handling and the treatment of infectious waste. Contingency plan should be developed to handle accidents that may occur. So infectious waste does have a different category. Okay, waste requiring special consideration. Mixed waste. Mixed waste consists of material that exhibit multiple hazardous properties such as infectivity, radioactivity, and chemical toxicity. Frequently, some of the components are strictly regulated by environmental protection and transport agencies of the local, state, and federal governments. However, treatment of disposal strategies for mixed waste generally are not addressed by most regulatory agencies. One difficulty, in part due to conflicting regulations, is that mixed waste frequently is not accepted at disposal facilities, operating under permits issued by Environmental Protection Agency or licensed Nuclear Regulatory Commission. Waste managers should anticipate that disposal problems will occur. Also, the concerns of the public regarding hazardous waste disposal in general is likely to influence the development of more restrictive mandates of handling mixed waste. So, such kind of waste should always be handled with care, and everything should be handled carefully, and care should be taken to avoid contamination of equipment, autoclave used for the process. When the uh, waste is being handled, biohazard symbol, universal biohazard symbol should be placed on the material, and whenever you are handling the radioactive waste, handle it according to the protocols, and uh, all those waste materials should be treated nicely and according to the protocols. Excreta that contain radionuclides from patients undergoing diagnostic nuclear medicine procedures may be discharged into sanitary sewer if the activity levels in the sewage do not exceed the license limitations. Excreta from patient receiving chemotherapeutic drug may be discharged into the sanitary sewer. If this practice is in compliance with local regulations, specimen of blood and body fluids obtained from these classes of patients often are submitted for laboratory analysis. These diagnostic specimens can be discharged into uh, the sanitary sewer, untreated or autoclaved, and disposed as a general waste. Chemical decontamination of mixed waste requires an assessment prior to treatment. Avoid the potential occupational hazards or difficult disposal problems. For example, decontamination of mixed waste uh, containing radio iodine. Incineration of mixed waste is very problematic, is dependent upon the regulatory requirements and permits for the hazardous chemical or radioactive components. Volatile metals such as mercury should not be incinerated but may easily be treated by other methods. Waste management strategies are hindered when the mixture contains volatile metals in addition to other hazardous waste components that uh, dictate incineration as a treatment of choice. Treatment of all regulated waste and regulated components of mixed waste is restricted to facilities holding the necessary permits. At the time of this lecture, all those thus mixed waste containing any dioxin group compound is difficult to manage due to lack of permitted facilities and resulting requirement for long-term storage at the producing institution. Stringent waste minimization strategies should be implemented to minimize the generation of this type of mixed waste. Human cadavers and anatomical and other anatomical waste. So, all anatomical waste require special handling and packaging due to, in part, to its putrescent properties at ambient temperatures. So, human cadavers or human dead bodies, they do have a tendency to putrefy. Cold storage is necessary for the non-preserved anatomical waste to minimize odors uh, and leakage problems. Human body parts, body and cadavers should be disposed of by the cremation, burial, incineration as stipulated in the regulation promulgated by the State Anatomy Board. Biomedical laboratories and veterinary research operations also generate anatomical waste such as pathological specimens and animal carcasses. Studies of animals and human infections may generate infectious anatomical waste. This waste may be packaged in a durable, preferably opaque plastic liner that is placed in the sturdy paper, fiber box or jar. As a precaution, absorbent material can be added to contain any fluids that might be present. Incineration is the treatment of choice for anatomical waste derived from infected host. A facility with a properly designed and operated medical waste incinerator can effectively treat infectious anatomical laboratory and veterinary waste for disposal. So here in this case, you can see the human cadaver. and how the experts are working on it. And such things are much more uh, exposed to the putrescent procedures at the ambient temperatures also. Similarly, you should always treat animal bedding in the similar manner and deal with it. Uh, the process of incineration and sharps. Whenever you are dealing with sharp objects, you should always uh, place them and store them in hard plastic container or orange color containers and having biohazardous symbol over that. Hypodermic needles, surgical blades are packaged in the disposable buckets made from high strength temperature resistant plastics. So used disposable needles and syringes should be placed intact and directly into the waste receptacle without recapping. Other safety design features may include a sturdy needle, sturdy handle to transport the waste safely, a restricted opening with self activating flap keep the waste covered while in use, and sealed the lid and contain the waste treatment during the disposal. So placing the waste receptacle in a location convenient to the activity generating the waste uh, is one strategy to ensure proper segregation and packaging of the waste to minimize uh, the risk of injury of housekeeping personnel. Coming on to the MCQ, which of the following practices should be utilized when working in a biological safety cabinet? A. Disinfectant. Disinfect the work surface for BSc before and after work. Disinfect all items which go into and come out of BSc. Do not store any items in BSc. Allow the BSc to operate before work begins and after work ceases, or all the above. Which of the following practices are not allowed in the laboratory? Eating and drinking, applying cosmetics, handling contact lenses, or all the above. It is important to wear two gloves and carry loose tubes in the elevator. True or false? When it is hot outside, it is okay to wear shorts and sandals to work in the laboratory. True or false? UV light can be utilized as the sole form of decontamination in a biological safety cabinet. True or false? Hands should be washed before and after working in the biological safety cabinet. True or false? Which of the following item is considered sharp? Needles, scalpels, microscopic slides and cover slips, or all the above? A laboratory working with human cell lines must display the biohazard symbol on areas where the work is performed or materials are stored. True or false? Some of the differences between a fume hood and biological safety cabinet (BSC) are that a BSC protects both the users and the materials inside the cabinet, and that the exhaust is HEPA filter. True or false? PPE is personal protective equipment, public protective equipment, possible protective equipment, or all the above. These are the references from where we have collected the material. And thank you very much for listening to my lecture.